Hello guys, hope you are doing well. Today we will start a new chapter. It's a chapter 18, regulation of gene expression in eukaryotes. Okay, so page 518 in your book. So كلنا بنعرف guys, already I explained this information in the first handout regarding the regulation of gene expression in prokaryotes. اتفقنا انه for any cell to respond to any change in the environment ورح زيد عليها كمان if any cells need to differentiate, so these cells must regulate their gene expression by turning on or turning off the expression of these genes in a coordinated manner. So in this chapter, we will focus on the regulation of gene expression in eukaryotes. So let's start by a brief introduction, guys. مثل ما بتذكر أنا وياكم our previous chapter in the handout lac operon, we learned about the process of regulation of gene expression in prokaryotes in bacteria. ومثل ما بتذكر أنا وياكم the structure of the lac operon كان عندنا a sequence known by promoter followed by a controlling site or operator and followed by 3 genes lac Z, lac A and lac Y and finally a terminator. إذا بنتذكر guys كان في عنا a repressor produced and this repressor will bind to the controlling site or to the lac O or also known by operator, preventing the binding of the RNA polymerase to the promoter site, and this binding of the repressor will block the RNA polymerase, preventing the transcription of the genes Z, Y, and A. So إذا لاحظتوا guys, the repressor will prevent the transcription. معناتها at the level of in the case of regulation of gene expression in bacteria or prokaryotes, the regulation will occur mainly at the level of the transcription. So أهم level بقدر أنا أعمل فيه regulation control للgene expression in prokaryotes or bacteria is the level of the transcription. So يا أما you will turn on the transcription and you will produce proteins, أو من الأساس you will turn off the transcription and you will not produce any kind of protein. Okay? طيب. اتفقنا إنه in this process guys, in this regulation, we need 2 important sequences, the promoter, since the binding site of the RNA polymerase, and the operator or the regulatory sequence. It's a binding site for regulatory proteins, such as the repressor in this case. But you have to keep in mind, guys, that regulatory proteins are not only repressors. Also, you can find other types of regulatory proteins, such as activators. So إذا حيلا كيس أخذتوه تاني غير اللاك أوبيرون وشفتوا في a regulatory protein such as an activator, in this case the activators will enhance, will stimulate the process of transcription. وهذا اللي رح نشوفه أكثر شيء عند اليوكاريوتس, because the eukaryotic transcription is regulated mostly by activators. In contrast, the prokaryotic transcription is regulated mostly by repressors. بس أكيد ما تنقول mostly يعني في عنا الاثنين, 2 types of regulatory proteins in eukaryotes و 2 types of regulatory proteins in prokaryotes, both activators and repressors. In addition guys, كلنا بنعرف إنه there is variation in the complexity of the regulatory systems among eukaryotes. كلنا بنعرف إنه the structure of the eukaryotic cells is more complex than the structure of a prokaryotic cells. And due to this variation in complexity, أكيد the process of gene regulation, expression of gene regulation, أكيد will also differ between the prokaryotes and eukaryotes. انتوا بتتوقعوا وين الاصعب؟ اكيد وقت نحكي about eukaryotic cells اللي هي considered as more complex, يعني اكيد وهذا الشيء منطقي انه نقول انه we have more levels for the regulation of gene expression in eukaryotes. Let's now compare the eukaryotic regulation with the prokaryotic regulation of gene expression. أكيد في عنا some points are common, so we will start by studying the key similarities, and then we will describe the key differences اللي هن أكيد more specific for eukaryotic cells, since eukaryotic cells are more complex than prokaryotic cells. So let's start first by the key similarities, the points shared by both eukaryotes and prokaryotes. What is common? First, the first common or similar point is the presence of promoter sequences. In both cases, prokaryotic and eukaryotic cells, we need the presence of a promoter sequence, which presents the binding site for an RNA polymerase. The promoter sequences also determine the start site, the +1 start site where the transcription starts. And also the promoter will help the RNA polymerase to distinguish between the template and the non-template DNA strand. And finally, the promoter also will specify the rate of transcription initiation. So the لاحظتوا promoter sequences have different functions. They are very important for the initiation of transcription. In addition to the promoter sequences, also a common point present is the presence of regulatory sequences. Regulatory sequences represent DNA sequences, okay, such as the operator in the case of lac operon in bacteria. كمان بنلاقي regulatory sequences بالcase of eukaryotes representing DNA sequences binding sites for regulatory proteins such as activators and repressors. So here we covered everything common between eukaryotes and prokaryotes, so promoter, regulatory sequences, and regulatory protein. هلا key differences. ليش عم نحكي عن key differences؟ لأنه كلنا بنعرف إنه the key differences will result from the greater organizational complexity of the eukaryotic cell. The eukaryotic cell is more complex than the prokaryotic cell. So خليني أعدلكم إياهم. هن in this slide they are represented by 5 points. وmainly هول الpoints اللي رح نحكيهم in this chapter. Okay? So the first key difference is the role of chromatin structure in regulating gene expression. شو معناتها guys? If we look at the structure of the DNA in eukaryotic cell, you can find that this DNA sequences are present in either a 10 nanometer chromatin fiber or 30 nanometer chromatin fiber. إذا بتتذكروا, we discussed in chapter 2 the differences between 10 and 30 nanometer chromatin fibers and how they can affect the process of transcription. When our DNA is present in its 10 nanometer chromatin fiber, معناتها the structure of the DNA is present in a decondensed loose structure. معناتها the transcription machinery can reach easily the DNA and start the transcription. However, when you change the chromatin structure from 10 nanometer to 30 nanometer chromatin structure, معناتها the DNA will be more packed, more condensed. The transcription machinery cannot reach easily the DNA sequence, so the transcription will be turned off. So the لاحظتوا هون guys, role of chromatin, how can affect the expression of the gene. Condensed معناتها gene will be turned off. Decondensed معناتها the gene will be turned on. Okay, so هاي أول point. The second one, أكيد we have an important point is the transcription, بس اتفقنا transcription is common between prokaryotes and eukaryotes. So ما رح نحكيها بالkey difference. The second point اللي هو كمان specific for the eukaryotes. After the process of transcription, a precursor mRNA will be produced, primary transcript. But this primary transcript or pre-mRNA cannot be translated in the cytosol or cytoplasm of the eukaryotic cell. First, it should be processed in order to form a mature mRNA. شو معناتها processing of the messenger RNA? Processing means the addition of a 5' cap at the 5' end of the messenger RNA, addition of the 3' poly-A tail at the 3' end of the messenger RNA, and finally, splicing of the messenger RNA means removal of introns and the ligation of exons. So هول هن ال 3 post-transcriptional modification and processing. هول modification بنعملهم للprecursor mRNA لنقدر نحولها to a mature mRNA, and now this mature mRNA can exit the nucleus into the cytoplasm. So this point is very important. If you missed the 5' cap, or if you missed the 3' poly-A tail, or if you didn't splice the messenger RNA, the messenger RNA will not be transformed into mature mRNA. معناتها you will stop the process of gene expression at this level. In addition, guys, important, very important point related also to eukaryotes is known by the alternative splicing. So the eukaryotic cells have the possibility to produce different messenger RNA from one precursor mRNA by a process known by alternative splicing. شو معناتها alternative splicing, guys? If we look at this structure, guys, it's the precursor mRNA. Let's consider that we have 4 exons: red, yellow, green, and blue, representing the 4 exons. By the process of alternative splicing اتفقنا انه we need to remove introns. But also sometimes, not only you can remove introns, but also you can remove some exons. Example, in the first part here, guys, we remove the 3 introns present between the 4 exons, but also the cell remove the green exon and maintain only 3 exons, red, yellow, and blue. The same cell, by alternative splicing and starting from the same precursor mRNA, will not only remove the introns, but also will remove the yellow exons, producing a mature mRNA with only red, green, and blue exons. So after translation, these messenger RNAs produced by alternative splicing will produce completely different proteins. These proteins are known by protein isoforms. They have completely different functions. They are represented as tissue-specific proteins. And in each tissue, you can produce specific type of this proteins. But please keep in mind, these proteins are produced from a single gene or from a single precursor mRNA. But how we can produce such different protein isoforms? By the process of alternative splicing. So that's why you have to keep in mind, guys, that from one gene you can produce different proteins, and this can explain why we have more proteins in our body more than the number of genes. Okay, so this is an explanation. In addition, guys, let's say انه كل هول البروسيسز عملناهم كلهم، خلصنا كل هيدا البروسيس, we produced the mature mRNA. Now it's time to transport this mature mRNA from the nucleus to the cytoplasm. تخيلوا guys هون at this level, at the level of the nuclear pore complexes, if you turned off, إذا سكرنا هيدا الباب، سكرنا this pore, messenger RNA ما بقى فيها تظهر, and you cannot produce the protein anymore. So this is another level where you can control the expression of genes in eukaryotes. Then, once in the cytoplasm, this messenger RNA will be translated into a protein. So also you can regulate or control the process of translation. So guys, please keep in mind this key similarities and key differences between prokaryotes and eukaryotes. So guys, in this chapter اتفقنا انه we will focus on the regulation of gene expression in eukaryotes. ورح شوف انا وياكم انه this regulation will occur at different various levels. But please listen carefully شو بدي اقول. Please listen, okay? We have to keep in mind, guys, that the regulatory needs of prokaryotes and eukaryotes are different. اسمعوا guys. The prokaryotes grow and divide. This is the normal life cycle of prokaryotes. Growth, division, growth, division. Whereas eukaryotes, guys, in their normal life cycle, other than the growth and division, we have the process of development and differentiation. أيها أصعب, guys, to simply grow and divide or to develop and differentiate? أكيد it's the second one. Eukaryotes develop and differentiate. This process is more complex than simple growth and division. So that's why eukaryotes require more sophisticated control of gene expression in space and time. عظيم. So اللي حكيناهم أول part كانوا introduction. So now we will start more to describe the different paragraphs of this chapter, starting by the levels of control of gene expression in eukaryotes. So رح شوفهم أنا وياكم ارجع in details من شوي. شفنا هيك brief عنهم بالintroduction while comparing eukaryotes with prokaryotes. So خلينا نشوفهم هلا in details ونشوف كل الستبس اللي رح نحكيهم in this chapter. So اتفقنا in prokaryotes in bacteria, the cell لأنه هي it's unicellular, so can respond quickly or rapidly to any change in the environment by turning off the expression of the gene, okay? And if any messenger RNA is present in the cytoplasm of the prokaryotic cell, it will be degraded rapidly, and when the cell divides, this messenger RNA will be diluted. In contrast, in eukaryotes, أكيد اتفقنا إنه it's more complicated than prokaryotes. أول سبب it's the compartmentalization. شو يعني the compartmentalization, guys? Means the presence of compartments, of organelles, of nucleus inside the eukaryotic cell. كلنا بنعرف in eukaryotes, the process of transcription will occur inside the nucleus. However, the process of translation will occur in the cytoplasm. However, in prokaryotes, both transcription and translation are coupled and occur within the cytoplasm of the prokaryotic cell. بينما باليوكاريوتس لأنه أصعب بكتير ولأنه كل وحدة مفصولة عن الثانية. So لأنه they are separated رح نشوف different levels where the transcription occurs. Okay, so there are more levels at which that the expression of genes can be regulated in eukaryotes. These are the 6 different levels where you can control the expression of a gene in eukaryotes. Starting first by the role of chromatin, then you can regulate the transcription, then you can regulate the processing, also known by post-transcriptional modification and processing. After the production of the mature mRNA, you can regulate the transport from the nucleus to the cytoplasm. So you can regulate the pores here at the level of the nuclear envelope. You can control, guys, the level of translation. So the messenger RNA, yeah, it will be stored or translated directly or degraded. And finally, the protein produced also can be processed, so you can regulate the level of protein processing. Protein processing, guys, means addition of sugars, addition of phosphate, addition of methyl group. Okay, so these are the processing of proteins. And the finally, It's the protein processing and degradation. So you can destroy or degrade the protein. وهاي هي آخر level where you can control the gene expression. إذا ما عملت control على هيدا الليفل اللي هو the level of proteins والبروتين صارت fully functional وراحت على قلب الكومبارتمنت محل ما لازم تشتغل, at this level you cannot control anymore the expression of this gene. Okay? So هول هن the 6 levels. We will discuss in this chapter the first 4 levels. Okay, 1, 2, 3, 4. And we will not discuss, no, we are limited in time, the 5th and the 5th and the 6th levels of gene regulation of gene expression. Okay, so we'll focus on these 4. And we will start by the transcription, then we will return back to the role of chromatin. عظيم. So رح نبلش بالtranscription. كلنا بنعرف guys إنه the process of transcription is divided into 3 steps: initiation, elongation, and termination. انتوا برأيكم guys وين الأهم إنه وقف الtranscription at the level of the initiation or at the level of elongation or at the level of the termination? أيها أقل ضرر؟ أيها which level will consume less resources and energy? أكيد if we stop the transcription at the level of initiation. So أنا ليش بدي أصرف energy, أصرف materials, أصرف products inside the cell ومشي الترانسكريبشن initiation elongation واخر شيء وقفها على termination من الاساس. So starting from the first step of transcription initiation, we need to regulate the process at the level of the initiation. Okay? So the regulation of protein coding gene expression in eukaryotes is mostly at the level of transcription initiation. لنحكي عن transcription initiation, we need to describe first the structure of a gene. What is present at the level of the gene, guys? So any gene is divided into different parts. So here at this level, guys, after the +1 start site where the transcription starts, here you can find the coding segment. So the segment which produced the messenger RNA and later the protein. But upstream, يعني قبل the +1 start site, we have important regulatory regions. شو في عنا؟ First, we have a promoter. Okay, so خلينا نقول إنه initiation of the transcription of gene is under the control of the promoter. Where is the location of the promoter? The promoter is present immediately upstream of the gene. شايفينه دغري وين الcoding segment بنلاقيه قبله. So upstream of this gene. But not only you can regulate the initiation of transcription by promoter, also we have another sequence known by enhancers. These enhancers, guys, that are distant from the gene. لاحظتوا منا بلزق الجين، شايفين حاطين هون فراغ يعني بعيد كتير عن الجين. So these enhancers that are distant from the gene are present either upstream of the initiation site or downstream of the initiation site. شايفينها guys؟ يعني يا اما بتلاقوا البروموتر هون وقبله upstream بنلاقي الانهانسر، او بتلاقوا البروموتر وبعده بنلاقي الانهانسر، بس ما بيهم. الانهانسر it's an important regulatory DNA sequence also affecting and regulating the initiation of transcription of a protein coding gene. So خلينا نحكي عن كل part لحد. Let's start by the promoter. Promoters are divided into 2 types. The first one is known by core promoter. شو يعني core promoter? The core promoter represents the minimal يعني أقل شيء the minimal portion, okay, of the promoter required to properly initiate transcription. So inside this core promoter رح لاقي important elements needed for the initiation of transcription. شو هن هال important elements? You can find the initiator first here, where you can find the +1 start site, but also you can find here at this level a sequence known by Tata box. The Tata box, guys, it's a consensus sequence, very, very important sequence required for the binding of important proteins هلا رح نحكي عنهم بعد شوي. In addition to the core promoter, قبل منه upstream to the core promoter, you can find a proximal element promoter. This proximal element promoter, guys, it's a sequence upstream of the gene that tends to contain primary regulatory elements. So here you can find a DNA sequence where regulatory proteins can bind. And finally, we have the enhancer. The enhancer مثل ما اتفقنا, it's present at a certain distance from the gene. بس من اسمها, guys, إذا بتسمعوا enhancer means this sequence will enhance, will stimulate the process of transcription for the maximal level. Okay, so the enhancer will stimulate, will push, will initiate, will stimulate or will enhance the process of transcription 100-fold. يعني عم نحكي maximum level. مين بيقعد على كل وحده من هالسيكونس؟ على الكور بروموتر guys you will find the basal transcription factors يلي بنسميهم كمان general transcription factors. هلا بنحكي عنهم بعد شوي. And also the RNA polymerase will bind to this core promoter. However, at the level of the enhancers and proximal element promoters, you can find the presence of specific transcription factors, not the basal one. We have specific transcription factors, the regulatory proteins, such as activators or repressors. The activators mainly will bind to the enhancer لأنه they match with each other. Enhancer means will stimulate the transcription. The same function will be applied to the activators. So البروتين اللي هن activators رح يجوا ويقعدوا على sequence of the DNA اللي بنسميها enhancer. كمان رح يكون عندنا regulatory proteins فيهم يكونوا activators or repressors كمان بيجوا وبيقعدوا على DNA sequence اللي بنسميها proximal element promoters. And these interactions between proteins and DNA sequence can initiate the process of transcription. So guys, رح ناخد 2 examples. So هون at the end of this video رح نشوف 2 examples. رح نشوف one example related to the regulation of transcription initiation by activators. ورح نشوف another example بس رح يكون regulation of transcription initiation by repressors. Okay? So we'll start by the activators. كلنا بنعرف guys إنه in order to initiate the transcription and then not only to initiate but also to activate يعني to the maximum level, the transcription initiation requires 3 classes of proteins. The first important class is known by general transcription factors, the basal transcription factors, بنسميهم كمان general transcription factors. You can find 6 different general transcription factors known by transcription factor 2D, TF for transcription factor 2, because we are discussing the RNA polymerase number 2, which is responsible in the production of the messenger RNA, finally leading to proteins. And these letters representing the types, okay, of the transcription factors. So we have 6 different types known by D, A, B, F, E, and H. الستبس اللي بدنا نقطع فيهم guys أنا حطيتهم by order, correct order. So خليني أشوف أنا وياكم how this general transcription factors will help in the initiation of transcription. First, this is the DNA guys, and this point represents the +1 start site. Before the +1 start site upstream, you can find the core promoter where you will find the data box. At this level, guys, في عنا the first general transcription factor known by TF2D. The TF2D has one subunit known by TBP for data binding protein. معناتها شو شغلتها هالبروتين؟ إنه تتعرف على data box. So the data binding protein اللي هي part of the transcription factor 2D will bind, so first will recognize and then bind to the data box. So مين هو أول general transcription factor بيجي على data box is the transcription factor 2D. Next, the transcription factor 2D will recruit the transcription factor 2A and transcription factor 2B. So صار عندي على الداتا بوكس 3 transcription factors اللي هن D, A and B. These 3 general transcription factors will prepare the data box, will create like a platform مثل أرضية, okay? لتقدر تستقبل عندها the RNA polymerase 2 attached to the transcription factor 2F. And finally, we still need the transcription factor 2E and the transcription factor 2H to join these complex, and at the end, with these 6 general transcription factors and the RNA polymerase 2, you will form the pre-initiation complex, also known by the complete transcription initiation complex. This complex alone, guys, اللي هن general transcription factors plus the RNA polymerase 2, can start the process of transcription, but at a very low basal level. Okay, so low basal level. These factors by themselves cannot influence the rate of transcription initiation. However, if you want to stimulate the process of transcription initiation, in addition to these factors, بدنا نزيد بعد عليهم the activators, because the activators من اسمهم will stimulate the process of transcription. So the first class of proteins required is the general transcription factors. However, the second class is the activators. The activators, guys, مثل ما اتفقنا, will stimulate the process of transcription initiation. إذا نطلع على shape of the activators proteins, رح نشوف إنه these activators proteins have 2 key domains. 2 key domains. The first one is the DNA binding domain, where the DNA will bind, where the protein will bind to the DNA strand. And also we have a transcription activation domain. لأنه it's an activator, يعني بدها a domain to activate the transcription. The scientists discovered that the activators are present as monomers. Monomers means composed of 1 subunit, or they could be present as dimers, يعني composed of 2 subunits. If the 2 subunits are identical, معناتها they are homodimers. And if these 2 subunits are different, these monomers are known by heterodimers. We will focus, guys, on this DNA binding domain. The scientists discovered that this DNA binding domain presents some common structural motifs. يعني إذا أنا بتطلع على حيلا activator بتطلع لهم على ال DNA binding domain, رح نشوف في عنا motif structures عم بيكونوا common between all these activators. Examples of these common structural motifs: helix turn helix, zinc finger, and leucine zipper. إذا بتطلع على الشيء تاعيتهم, guys. So this is the first one, helix turn helix. And this is the DNA, representing the DNA double helix with minor and major groove. Because of this specific shape, guys, اللي هي helix turn helix, similar to a hairpin, مثل دبوس شعر, this structure can fit in the major groove of the DNA double helix. So this is the part of the activator that can bind to the DNA. The zinc finger, guys, it's similar to a finger with a zinc moiety in the middle, and this represents the recognition site of the helix by which recognize the DNA. So هون بيكون عندنا a perfect fit between the zinc finger and the DNA. So هيدا كمان it's one important common structural motif of the activator, specifically the DNA binding domain of the activator. And this is the leucine zipper. It's like a zipper, مثل السحاب, guys, where you can find here in the middle leucine, leucine, leucine. And this shape can also fit or bind to the DNA. So حكينا, guys, عن the DNA binding domain of the activators عندهم these common structural motifs. وعندنا activation domains, but the activation domains are not common. You can find a variety of forms of shapes specific for each activator. The presence of the activator, guys, will stimulate the transcription initiation up to about 100-fold. Without the activator, you can start the transcription with the pre-initiation complex, but at a very low basal level. I will present here, guys, I will present the structure how the DNA will recruit all these factors discussed previously. ومنشوف كيف بيصير في عنا الانتراكشن between all these factors. This is the structure of the gene, the coding segment, the promoter with the theta box, and the upstream enhancer. So the activators will bind to the upstream enhancer, the transcription factors, general one, will bind to the theta box, and this is the RNA polymerase. إذا بنجي بنطلع guys على هيدي الستراكشر رح تشوفوا إنه the DNA شايفين هيدا DNA guys السيكونس اللي هي between the enhancer and the promoter هيدا البارت اللي هون. The DNA containing the enhancer is brought close to the promoter. شايفينها guys عم بتلف. So this DNA with the enhancer sequence is brought close to the promoter side, to which the transcription machinery is bound, stimulating the transcription to the maximal level for the particular gene. So this is another representation, gene مع البروموتر مع الانهانسر. الانهانسر كتير بعيد. طب إذا قاعد هون activator, how this activator will contact the general transcription factor and stimulate the transcription. ليصير في direct contact between the activators from one side and the general transcription factor and RNA polymerase from the other side. This DNA in the middle will fold on itself, تبرم على حالها. هيك الانهانسر وين صاروا guys facing the promoter، وهون the proteins attached to the enhancer and to the promoter رح يكونوا in direct contact, and in this case the activator can stimulate the transcription to about 100-fold. So شفنا guys, for the regulation of the transcription initiation by activators, we need 3 classes of proteins. The first one was the general transcription factors, the second one activators, and the third class of proteins are the co-activator. الco-activators guys مثل ما كنا نقول مثلا co-enzyme, okay, co-factor. Co means will help the activator to perform a certain function. The co-activators guys مثل ما شايفينها هون بالنص, it's a very large multi-protein complex. But the co-activators will not, okay, bind directly to the DNA. The co-activator will bind first to the activator from one side and to the general transcription factors and RNA polymerase from the other side. Okay, so there is no direct interaction between co-activators and DNA sequence. The co-activator is present in the middle between the activator and the general transcription factors and RNA polymerase. However, the activator will bind directly to the DNA because the enhancer, it's the DNA sequence. Okay, so the activator هخبركم step by step guys how the recruitments of all these proteins will occur. So step 1, activator will bind to the enhancer, then recruits a co-activator. the co-activator will recruit the RNA polymerase 2 with the general transcription TF2F. At the same time, at the level of the Tata box, the transcription factor 2DAB will bind, recruit the RNA polymerase 2 اللي هو already attached to the co-activator activator, and finally in order to complete this complex we need the transcription factor 2E and the transcription factor 2H. وهيك بيصير عندنا complete, it's not the pre-initiation complex, it's the complete initiation complex ready to stimulate the process of transcription to about 100. So this interaction مثل ما اتفقنا between activators, co-activator RNA polymerase 2, RNA polymerase 2 general transcription factor will stimulate the transcription initiation to about 100. In addition, guys, the scientists discovered that many co-activators also contain enzymes. These enzymes can modify the chromatin structure. بأي معنى؟ If the DNA is present in the 30 nanometer chromatin fiber, معناتها condensed form, this co-activators with enzymes will open the chromatin from 30 nanometer to 10 nanometer. The general transcription factors, the RNA polymerase 0, more accessible the DNA, where the transcription will occur. So that's why we need to change, to modify the chromatin structure before the initiation of transcription. And one kind of co-activator is known by mediator. Mediator هو co-activator, but it's found originally in yeast. So guys, please, كل ما تسمعوا the term mediator, it's an example of co-activator, but found in yeast. So this is the structure, guys. So this is the mediator. It's found in yeast. شوفوا قد ايه it's a very large multi-protein complex composed of 20, at least 20 polypeptide. So إذا بتلاحظوا, at the level of the enhancer, the activators first will bind, then recruit the co-activator mediator. The mediator will recruit the RNA polymerase 2. The interaction between the mediator and the RNA polymerase 2 will occur mainly at the carboxyl terminal domain CTD, اللي هو الطرف آخر طرف من ال RNA polymerase بيظهر منها like a tail at the C terminal end, بنسميه carboxyl terminal domain. وهون بيكون عندنا at the same time at the Tata box the transcription factor 2D will bind and recruit the other general transcription factor, and the interaction between all these proteins will stimulate the transcription to about 100. Finally, the inhibition of transcription initiation by repressor. So this is another kind of regulatory protein. شفنا الactivator, هلا بدنا نشوف الrepressors. The repressors, when they are present, will inhibit the activation of transcription initiation by activators. These regulatory proteins known by repressors, structure-wise, are similar to the activators. So also they have two domains, a DNA binding domain, and the repressors can bind to the DNA sequence. And here we need a repression domain, and it's a repressor. بينما بالactivator كان بدنا DNA binding domain وزيادة عليها كان بدنا an activation domain. So how the eukaryotic repressors work? In different ways, we will describe 3 of them. In one way, مثل ما شايفين بالfigure guys هون, the activator binding site overlaps the repressor binding site. يعني هول التو sequences, وحدة for the activator وحدة for the repressor, بس their sequences overlaps يعني بيجوا فوق بعض. So if the repressor binds first, the repressor will prevent the activator from binding, وهيك you will inhibit the activation of transcription initiation by activator. طريقة تانية, in another way, إذا ملاحظين the binding sites are different. Activator will bind to the activator binding site, repressor will bind to the repressor binding site, but through interaction of the repression domain with the activation domain, the activator action will be blocked. So رح يصير في interaction between the activation domains اللي هن أول وحدة repression domain والثانية هي activation domain. So this interaction مثل كأنه repression domain will mask or hide the activation domain, and at the end the activator's action will be blocked. And finally, alternatively, some receptors will recruit co-receptors. كيف قبل كنا نشوف some receptors will recruit some activators will recruit co-activators. هون نفس الشيء some receptors will recruit co-repressors, وهيك you can also turn off, repress the transcription initiation. So that's it, guys. Thank you for listening. If you have any question, please let me know.